Welcome to the interactive video presentation on the use of selective and differential media. In this video, you will be watching a demonstration of the use of selective and differential media for the detection and identification of microorganisms. General purpose media, example plate count agar, is used generally for performing microbial counts. This media contains nutrients to allow the growth of most bacteria and fungi. A total count of all growth is taken using this agar. Selective media is used where identification of the bacteria is required. It contains similar nutrients to non-selective but contains agents to either encourage certain bacterial strains and or suppress others. Rose Bengal chloramphenicol agar, for example, contains an antibiotic, chloramphenicol, to suppress bacterial growth and a dye which is selectively absorbed by yeast, Rose Bengal. It is important to count only those organisms showing characteristic growth and reaction for that agar. Other bacteria or fungi may still grow but should not be counted. Differential media, on the other hand, is used to differentiate between different organisms growing on the same plate. For example, mannitol salt agar is selective to Staphylococcus species. However, the media is used to differentiate between Staphylococcus aureus and Staphylococcus epidermidis. This media contains a pH indicator which turns yellow in the presence of acid. Staphylococcus aureus produces acid and therefore colonies appear yellow, as well as the media itself changing color from pink or red to yellow. Staphylococcus epidermidis, on the other hand, does not produce acid, so media remain pink or red and colonies appear pinkish or red in color. Where small numbers of bacteria are expected, or a possibility of the presence of fastidious organisms, an enrichment process may be required. This enrichment media contains nutrients to encourage the growth to detectable numbers. Samples tested for Salmonella species, for example, are incubated for 24 hours in enrichment broth prior to inoculation onto selective media. The objectives of this demonstration are to identify bacteria from growth and reactions on selective media, to understand cultural morphology and colonial characteristics. To achieve this, we will use a set of 7 microbial cultures including Bacillus cereus, Escherichia coli, or E. coli, Pseudomonas aeruginosa, Staphylococcus aureus, Staphylococcus epidermidis, Saccharomyces cerevisiae, and Salmonella typhimurium. 7 each of 9 different uninoculated selective and differential media plates including eosin methylene blue, Baird Parker agar, mannitol salt agar, Rose Bengal chloramphenicol agar, malt extract agar, Brilliance Salmonella agar, Brilliance Bacillus cereus agar, Pseudomonas selective agar, and chromogenic Listeria agar. 7 uninoculated McConkey broth. These are shown in the table. We will streak each organism onto one of each selective and differential media plates. First, label on the reverse side of the plate with the organism name. Mix each microbial culture thoroughly. Hold a metal loop at an angle in a blue Bunsen flame until it glows orange for a few seconds. Allow the loop to cool. A selected area on the agar surface, close to the edge of the plate, can be used to cool the loop. Take a loopful of microbial culture. Lightly streak on the agar surface to produce a streak plate. Refer to the interactive video on gram staining and microscopy for how to prepare a streak plate. Flame and cool the metal loop in between each plate, and when finished, repeat this for each type of selective and differential media for the 7 microbial cultures. In the case of McConkey broth, Label the bottles with the names of the microbe. Transfer a loopful of each culture into corresponding bottle of broth using a sterile loop. Before the transfer, ensure the inverted tube in the bottle is full of broth. To do this, ensure the cap is tightly closed. Turn the bottle slowly to the horizontal position, then slowly back to the upright position. The plates and broth are now ready for incubation. Incubate at respective temperatures and time before observation, as shown in table. After incubation, the plates are examined for any growth and morphology of colonies, as well as changes in media characteristics. The results are then recorded in the table. From the completed table, you will then be able to identify which media is selective for each organism. Examples of positive results of streaked plates of various selective and differential media are now shown. McConkey broth shows positive reaction to E. coli. The broth turns turbid, there is acid production and gas is evolved with is collected in the inverted tube. Color of the broth changes to yellow from purple. Eosin methylene blue is selective to E. coli. Isolated colonies are 2 to 3 mm in diameter, with little tendency to confluent growth. Colonies appear greenish metallic sheen by reflected light and dark purple center by transmitted light. Baird-Parker agar is a selective as well as differential media. It is selective to Staphylococcus species. On the other hand, it differentiates Staphylococcus aureus from Staphylococcus epidermidis. Staphylococcus aureus appear gray-black shiny convex colonies, between 1 to 1.5 mm in diameter. Colonies are surrounded by an opaque zone and a clear zone. Staphylococcus epidermidis, on the other hand, appear gray-black but not shiny and seldom produces clearing zone. Mannitol salt agar is another selective as well as differential media for Staphylococcus species. Mannitol salt agar also differentiates Staphylococcus aureus from Staphylococcus epidermidis. Staphylococcus aureus appear as yellow colonies with yellow halo. Media color changes from pink to yellow. Staphylococcus epidermidis appear as pink colonies with pink media. Malt extract agar is a selective as well as differential media. It is selective to fungi but differentiates between yeast and mold. Yeast appear creamy yellow or pink uniform round colonies on malt extract agar. Mold, on the other hand, appear filamentous in growth. However, color and growth vary depending on species. Rose Bengal chloramphenicol agar is another selective as well as differential media for fungi. Rose Bengal agar differentiates between yeast and mold. Yeast appear as pink-colored colonies. Mold appear as mycelia black spores growth, which vary depending on species. Brilliance Bacillus cereus agar is selective to Bacillus cereus, which appear as blue-green colonies. Pseudomonas selective agar, as the name implies, is selective to Pseudomonas species. Colonies appear straw-colored with green pigmentation. Brilliance Salmonella agar is selective to Salmonella species. Colonies appear purple in color. Klebsiella species enjoy good growth on this media, however, colonies appear blue-colored. Chromogenic Listeria agar is selective to Listeria monocytogenes. Colonies are blue-green in color, with clearing zone around colonies forming halo. Listeria innocuous show growth with no halo. The results are summarized in the following tables. To conclude this session, consider the following questions related to the use of selective media in microbiological detection and identification. You will have to do additional reading or research in answering them. 1. What is the main difference between a general-purpose media and selective media? 2. Explain the differences between differential and selective media, giving one example of each. 3. What is the meaning of the term enrichment as use in microbiology? When must enrichment step be included in a microbiological analysis? You have now reached the end of this demonstration. You may watch it again when needed.